Hello.
The Father, the Son, the Holy Spirit. Amen. God, grant me the serenity to accept the things I cannot change, courage to change the things I can, wisdom to know the difference. In the Father, the Son, the Holy Spirit. Amen. All right. So we got Jasmine, Callum, Eden. Emily, way over there. Abigail. Ashley. Up I know she is. Right. Anna. John. Curly. Quattro. Cameron.
Can you hear me
Back
right
there.
now? I'm online right now. My email wasn't working this morning for me to email you, but I am here online.
Okay, thank you. Rebecca. Rebecca. John Colst. Yo. Kate. Marie. Sam just came in. Sophia. Sophia. Christina. Kristen. Oh, there you are. Sarah over there. Tristan. Yo, what's up? Okay. Got Caleb right there. I see you. Got you, Caleb. Jordan. Yep, that's right. I saw you on the way in. Antonia, right there. Okay. There's Jonah. Landon. Okay, and some more. 2 more people. This is wonderful. Transport. Getting something across the plasma membrane. Two ways. Passive transport. It's going to move from high concentration to low. Doesn't require any energy. Just like dropping the hat. Once I let go of it, it's down. Water running downhill. Passive. Boom. Right? Easy. Easy. Active transport. All right. You have to put energy into it. Lifting the hat. To move substances, we said, up or against a concentration gradient. So we're going to see some examples of that later today. Basically, what that means is I'm moving from low to high concentrations. So you look at the numbers. So, active transport. If we're going to move sodium, first of all, quick review, does sodium easily move through plasma membranes? Yes or no? No. If it's charged, if it's polar, it doesn't easily. That's information. Got to know that. This is like a language. In or the. But anyways, so if it's charged, it doesn't move through. And we're going to say we're going to move this actively. So what direction are we going to move here in this case? From right to left or left to right? From right to left. Right? If it's active transport, it's going from low to high. Active transport, easiest way to define it. Anytime something moves from low to high, that's active transport. And then on this case, left to right, that would be active transport. Okay. So that's the general classifications of transport. Passive and active. Now we're going to look at specific kinds of each. Why do I have my hat in my hand? So diffusion. We need to know about diffusion. Simple diffusion, facilitated diffusion. These are both examples of passive transport. When you hear the word diffusion, passive transport. And Fick's law of diffusion, all these variables, all right, it basically defines how fast. So the rate, so think of rate, how fast something moves across a membrane over time. That's a rate. So Fick's law of diffusion tells me I can predict, based on different factors, how fast something's going to move across the plasma membrane, given different conditions, okay? So some of the main points. C2 minus C1. This C2 is the concentration outside the cell. C1 is the concentration of that same substance inside the cell. This is important. So, if we take and we just look at these numbers, okay, let's just use some simple numbers here. 24 millimole, 10 millimole. All right, we're not going to use units right now, but let's say that's the same unit. 24 is on the outside, 10 is on the inside. What's the result of C2 minus C1? Is it positive or is it negative? Quattro. Positive. So, when C2 minus C1 is positive, first of all, that tells me the direction this thing is going to move. When C2 minus C1 is positive, I have a positive concentration gradient. Gradient simply means difference in concentrations in two areas. It's going to move into the cell. C2 minus C1 now is going to be negative. If it's negative, we have a negative concentration for this particular molecule. All right. And there's different ways of doing this. Sometimes people put a negative in front of this whole thing. We're just going to keep it simple. Try to simplify the math here. We're going to say that we get a movement of X out of the cell. So it's really simple. If we had, let's say this door over here was about ready to open, and there was me standing on this side, and there were 80 people on the other side, and the door opened, what's going to happen? Or is it more likely for people to come in or me to get out? Jonah? It's more likely that there's going to be people coming in, okay? So unless they all stopped and said, oh, here comes here's the professor. But you got it. If we were playing a game and it was whoever gets to the other side of the door wins, I'm not going to win that, right? So this gives us an idea of which way molecules are going to move across the membrane. So concentration gradients. And the bigger the concentration gradient, let's say this difference gets larger. How does that affect the rate of the movement? It's going to move faster, right? Again, using the image of the door, the people moving in, if it's 80 to 1, will be much faster than if it was 80 to 60, right? Okay. Second thing, the property of the cell. Main factors here, let's look at the membrane thickness. It's in the denominator. So as the thickness of the membrane increases, what happens to the rate of diffusion? If the John? Right. And as the membrane thickness decreases, the rate of diffusion is going to increase. Oh, look at that. I actually have that. Okay. The other thing would be the surface area. As the surface area of the membrane increases, what happens to the rate of diffusion? Jonah? Okay, so surface area and rate have there's a direct relationship. Concentration gradient, the difference in concentrations of this molecule across the membrane, there's a direct relationship. What I mean by that is if one goes up, the other goes up. But the membrane thickness, it's what we call an inverse relationship. If thickness goes down, diffusion goes up. And as the membrane thickness goes up, diffusion is going to go down. That should make sense, right? It should make sense. If the membrane is thicker, it takes longer for that molecule to move through that membrane. So you should know you won't have to know this equation, but when this equation shows up on the test next Friday, you should know how to interpret it. Because it will definitely show up. Okay. Passive transport. We're going to look at three things. Simple diffusion first. Molecules that move across the membrane by simple diffusion will be small nonpolar and large nonpolar molecules. Oxygen and steroids. They can simply move through the membrane. So in order to increase the rate of the diffusion of those molecules into a cell or out of the cell, all you have to do is change the concentration gradient. You pump more oxygen into the blood, there's going to be more oxygen that then could move into your cells. Okay. Simple diffusion does not need any help. No transport protein needed. Okay, so that's another characteristic. Oxygen doesn't need anything to help it get across the membrane. It just goes. Whew. Oh, yeah, I just told you that. But I guess unless it's on the board, we don't. Anyways, so simple diffusion. Rate of diffusion determined by the concentration gradient. You don't need a transport protein. It needs no help getting across. Okay, facilitated diffusion. Facilitate. Somebody that facilitates something. What's another way of using that word facilitate? Aiden. Helper. Helper. So this is diffusion that needs a helper. What kind of molecules need a helper to get across the membrane? John? Polar and charged molecules. Boom. Good. So polar and charged molecules, they need a helper. I'll need a helper. Why not? Dun, dun, dun. That's the $1 million question. See if we remember from Friday's episode of plasma membranes. Christina, why can't they get through? What stops them? Do you remember? Inside the plasma membrane. Let's see. Yes. It's the nonpolar tails. So sodium tries to go in. It makes it through these polar areas. Okay. Doing, 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 doing. It hits the nonpolar, right? Hydrophobic. So if there's a molecule, really, really simple. If it's either water does not easily pass through. But if it's a molecule that can interact well with water, as soon as it hits the nonpolar tails, it hits this hydrophobic exclusion shield, right? Whew. Can't get through. Sweet. So these substances, they need a helper. They need a protein to help them pass through. That's why it's called facilitated diffusion. It's still diffusion. It's still going to move passively without energy, but it needs a helper. One example of a protein that can help move things, pores. I guess you want to call it a pore. They like to call it pores. Ion channels is a more accurate word. They like to use the word pore. So pore is like a protein hole through the plasma membrane that enables ions or polar molecules to move in or out of the cell. So when this pore, sodium channels, protein. What's the monomer for protein? Amino acids. Okay. Normally, sodium channels, there's a group of sodium channels that are normally closed. They're proteins. And in the right conditions, when the voltage changes, when there's an electrical voltage change, they all start moving. You know, way cooler than those sliding door thingies that you see in Star Wars and stuff like that. These are the originals. And this hole opens right in the middle of the membrane. It's awesome. So when the pores open, the sodium can move right through because it's open, and water can then move through here as well. Anyways. The pores are oftentimes selective. They allow particular molecules to enter or exit the cell. So if it's a sodium channel, sodium's going to move through. We're going to see a different kind of pore just for water. Okay, so if the pore is open, here we're talking about diffusion, so this is passive transport. We're gonna keep this really simple. We're gonna simply say it can move through if there's a concentration gradient and if there's an electrical gradient that promotes or would allow this to move through. So the inside of the cell, this is inside, by the way, on the right side is the inside of this cell. The left side is the outside of the cell. So if there's a pore here that opens, sodium can now move from right to left because there's a concentration gradient for it to move from right to left. Is there an electrical gradient for it to move from right to left? What I mean by that is with the charge on the inside of this cell, which is negative, would that attract a positively charged ion? Yes, it would. Okay. So the pore must be open. Then sodium can come in. So let's stop there for a second. Questions about that? Yep. It depends. Totally depends. It depends on what the particular kind of protein is. Anyways, yeah. So this example is showing beta sheets, but there are definitely channels where the main regions going through the membrane are those alpha helical shapes. Okay, good question. So, but I'm glad that you can see this was a beta sheet. An example of what level of structure in the protein? Secondary. Held together by what kind of bonds? Hydrogen bonds. Okay. So one way, anyways, that an ion can, or a charged molecule or a polar molecule can enter or exit the cell is through what are called pores. The second are, another way, are called carrier proteins. Carrier proteins. These things, they literally transport polar molecules like amino acids, sugars across the plasma membrane. They literally grab it and lug it across. So here we see, first of all, let's ask ourselves a question. See the little, what color is that? What? Purple. Sure. I'll accept that. I don't really accept that, but that's okay. Anyways, if it's purple to you, no, I'm just kidding. So air is purplish. So first of all, here we have our purple square structures, or diamonds, whatever you want to call them, up here. This is outside, and here's inside. So what's the concentration difference here? Outside it's, inside it's. So this thing is going to move into the cell. Because it's going from high to low, I know that it is passive transport. Because I'm using a protein, it is facilitated diffusion. Got it? Good. All right. So, first thing that happens is this molecule comes in. We're going to call this a sugar, even though it's not a hexagonal or a pentagonal looking structure. We're still going to assume it's a sugar. And it binds to a site on the extracellular side of this protein, and then it changes shape. So it interacts with those amino acids, and then they begin to change shape. The change of shape, literally, it carries it, it pushes it from one side to the other. That's what happens. It literally changes shape to move it from one side to the other. This process takes time. Whoops. So, this process takes time. So, of the two forms of facilitated diffusion that we talked about, one was moving through a pore, the other is moving through a carrier protein. One of these is much faster than the other. One of these things is faster than the other. Which one of these two is faster? Told you one takes a lot of time. So, anyone ever been? This is a very serious question. Sorry, this is a very serious question. Anyone ever been to like a big sporting event or a concert? Lots of people. What's the speed of getting in versus the speed of getting out? How do they compare? Getting in is it faster or slower than getting out? Slower. Why? John. What's that? Okay, security checkpoints. That would be like making sure it binds. Good. What's another thing? All right, I'm going to do some charades here. Ready? See how this works. What did I just do? Come on, what is that? You guys, turnstile? Did you guys ever go through the turnstile? Man, you guys need to learn how to play charades or act stuff out. That's bad class participation. So, in other words, it's slower because you can only go one at a time. Or two, yeah, you can't go two at a time. One at a time. Sometimes in these carrier proteins, they can transport 2 or 3 at a time. But each time it requires time, so it's slower. When they let you out of that stadium, they want you out. They open the gates, the big wide gates, especially like PNC Park and stuff. People are ripping out of there 5, 6, 7, 8 wide. Get me out. I'm going to get back to my car. So these carrier proteins, when you're transporting by carrier proteins, it's much, 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 much slower. All right? It's just something to keep in mind as we go forward. All right, finally, let's osmose. You're going to learn that by osmosis. No, you're not. The osmosis doesn't work that way. Well, maybe it does. I don't know. So, osmosis is a special kind of diffusion for water. It's a special kind of diffusion for water. It's still diffusion. It's still passive transport. Get lock and load. That's got to be locked and loaded, ready to go. Get that in there. Diffusion or osmosis is a specialized kind of passive transport for water. And the proteins, the pores which allow water to come in, are called aquaporins. It's a water pore. That's literally what the word means. Aqua, water, porin, pore. And diffusion of water occurs by osmosis. Now, instead of saying concentration gradient, we use the term osmotic gradient. Water moves across a plasma membrane through aquaporins according to osmotic concentration gradients. So the osmolarity of a solution is the number of solute molecules in a particular volume. So osmolarity tells me, what it directly tells me is we could say how salty is this solution. And that's what drives the direction of the water movement. Just said that. Now, here is where everything flips on its head. Water moves from regions of low osmotic concentrations to regions of high osmotic concentrations. It's not active transport, but that's how water moves. It's backwards, in a sense. So, yeah, you got this slide right here. Just scribble it. You can just sort of like scribble it out for right now. So you got 2 pots of soup. Pot on the left, very low salt, bland. Pot on the right, very high salt. Sometimes that happens here on occasion. Taste it. Woo. Maybe that's never happened to you. Happens to me sometimes. Okay. Low salt soup, very high salt soup. Not much taste, too much taste. So, here's your job, hotshots. You're going to move only water. How can you adjust the soup in each pot so that the saltiness of each pot ends up being the same? Murmur amongst yourselves for a few minutes. Murmur, 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 murmur. Moving only water. Do, 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 do. Do, 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 do. This is for all of you who are watching at home. I hope that you are enjoying my little Jeopardy. Probably, do you guys even know what Jeopardy is? Do you know what Jeopardy is? Did you ever watch it? Heck yeah. I haven't heard that all semester. I heard it all the time 2 years ago. Do, 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 boom, boom. Okay. How are we going to do it? What are we going to do? Simple. This is simple, right? We want to make. Okay, Jonah, you got your hand up. So that's, if we want to make the saltiness of the two soups the same, and this is a totally imaginary thing, right? Well, you could take this and you could run it through a distill. You could run it through a still. You could distill off the water, evaporate it over here, and whatever. Stop. I know that's what you were thinking, right? Because Quattro's a chemistry guy. He's like, man, I got this. Give me the equipment. I can make this happen, right? That's what you're thinking. Anyways, that's right. And as I remove the water from this side, what happiness? I like that word. What happiness to the saltiness in the left pot? Rebecca. Yes, so as I remove the water from the left side, what happiness is that the saltiness goes up here. And when I add the water to the right pot, what happens to the saltiness of the right pot? Jasmine. Does it go down or up? Yes, it becomes less. Actually, that time I was just looking for a simple direction. So it becomes less salty, right? So the saltiness is going to go down. That's simple. We do this all the time. It's called osmosis. That's how it works. So now instead of having low salt soup and high salt soup, we have some soups that have different osmolarities, because this is what you would actually look at. Milliosmoles per liter. That's how you, and then we have a little tube here. No, we are going to get into a little, is this chemistry or would this still be like biology? Yeah. Biology, okay, all right. We have a little tube that connects these two, a pipette with a little semi-permeable membrane, only allows water to move. Because ultimately, this is what we want to look at. The movement of water through an aquaporin channel across a plasma membrane. Does it go in or out? That's what we want to look at. Well, okay. So if this is the case, osmolarity is a way of saying how salty this solution is. So then the water would simply move across this plasma membrane, move from this pot to that pot, until the osmolarities are the same. That would be reaching a state of equilibrium, where we got the same in both sides. Okay, water moves from low to high osmolarity. And we could also say that this actually, if the osmolarity is lower here, then the water concentration is actually higher on this side, right? That's another way of thinking about it. Okay, so let's stop there for a second. Questions about that? Okay, nope, all right. Well, how does this affect me? Why do I care about any of this? This number is absolutely, this is one of those numbers that you need to emblazon, tattoo on your brain. The movie Memento, it's not like highly recommended on the monk scale, but it's a pretty cool movie. And he has short-term memory loss. He tattoos everything to his body, or he makes notes. This would be one of those things. If you have difficulty in, well, maybe you shouldn't, because then if I see you looking at your forearm, I think you're looking at your tattoo that has the answer. But 300 milliosmoles per liter. Put that in your memory banks. Inside of your body, that's the osmolarity. Inside of your cells. That's the goal. When the osmolarity gets outside of that, your body starts responding to make changes. So if we compare what's inside the cell, all right, here we can say inside the cell, maybe this would be our one of these soup pots. So inside the cell, it's 300. If the solution that's outside the cell is also 300, they're the same. We call that isotonic. Tonicity is another way of expressing osmolarity. Isotonic. Same osmolarity, inside and out. No net movement of water. That doesn't mean there isn't the movement of water, just there is no net movement of water. All right, now what if the osmolarity in the solution outside the cell is 280? And the inside is always going to be, we're going to say, to start out with, the inside is always going to be 300. Which way is water going to move? Into the cell or out of the cell? You can write it down on your notes if you want to, if it helps you like visualize it. Inside the cell it's 300, outside it's 280-ish, we'll say. Okay, well, first of all, if the osmolarity of the solution outside of the cell is less than 300, we call that hypo is below tonic. And it would move into the cell because it's going to move from low to high to make the saltiness of both the fluid on the outside of the cell as well as inside the cell the same. And the cell would then swell, expand. If the osmolarity outside the cell is 300, greater than 300, excuse me, like 310, we call that hypertonic. And water would do what? What would it do now? Kristen. It's going to move out of the cell. Right, cell will shrink. So that's osmosis. Questions about that? Think about the soup. Which way? The goal is to try to get those two to be, they're going to be the same. That's the goal when water's moving. Okay, all right. Now let's talk a little bit about active transport. So, requires the movement of energy to move substances against the concentration gradient. So from low to high, when I lift up, whoop, I lift up the hat. Okay? And we're going to look at 3 ways. And these are all going to use a transport protein, by the way. They're all going to use a protein. And in some ways, they're going to directly or indirectly utilize ATP. Okay? But the main way that you should distinguish if it's active or passive is what direction is it being moved? Is it being moved from high to low or low to high? That's the main characteristic I want you to focus on. So uniporter. So, and these tell us which way it's going to move. So uniporter, it's going to move one thing. It's like one of the, what's that famous saying that, well, maybe it's not so famous now, but it was 3 months ago, which means probably nobody uses it now. You have one job, right? So it simply moves one molecule, uses ATP to transport one molecule against a concentration gradient. Uniporter. Simple. Okay. Anti-porter. That's opposed to your Uncle Porter. Hey, that's a joke. Write it down. Thank you. Bam. On Wednesday, I'm going to saturate myself with kerosene before I come into class, and then I'm going to light myself on fire and see if you guys respond. Hey, there we go. That's good. Yes, it's my goal for the week. I got it. Okay, anti-porter. So it's going to transport one kind of molecule into the cell and one out of the cell. It's taking it different directions. Anti-porter. Whoa, example of this is the good old sodium potassium ATP ACE. ATP ACE. It's going to cleave a phosphate off of ATP. So ATP ACE, it's using ATP to make all this happen. And basically what it does, we see all these steps. Big picture here. I'm just looking for big picture here. Sodium potassium ATPase uses ATP to transport 3 sodiums out of the cell and 2 potassiums into the cell. Easy. Sodium potassium ATPase uses ATP to transport 3 sodiums out of the cell, 2 potassiums into the cell, and both of those movements are against concentration gradients. That's simple. That's an example of an anti-porter. Sodium potassium ATPase. One of the most common important transport proteins that we have in our bodies. Sodium potassium ATPase. It's what keeps your sodium concentrations outside of your cell high and your potassium concentrations inside your cell high. If it wasn't for this, if they weren't high and that wasn't high, you would die. Okay. Sym-porter, Sym, S-Y-M. S-Y-M. go in the same direction, same direction, same direction. Okay, you probably have this text. Uses energy of one molecule from regions moving from high to low to move another molecule in the same direction from low to high. So because there's going to be one molecule, at least one, that moves from low to high, that makes this a form of active transport. An example of this is the sodium glucose linked transporter. Sodium glucose linked transporter. So here's our little ATPase doing its thing over here, pumping potassium in, sodium out. Bup, 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 bup. Then over here we've got this little protein. It's going to move sodium from outside to inside. That's passive transport. But it's going to move also at the same time glucose from a low concentration outside the cell to a high concentration inside the cell. So this is a form of active transport because we're moving glucose from low to high. That's active transport. Technically, they call it secondary active transport, but I'm not going to worry too much about that. You can worry about that in cell or something. These proteins are really, 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 really critical in the kidney. When your kidney's reabsorbing, which means reclaims for itself what it needs, the glucose in the kidney. Okay. So, for Wednesday, I'm not going to do the kerosene thing, by the way, just in case you were really holding out hopes for that. For Wednesday, I'm going to be posting a little note on Schoology about a couple of just some videos that I want you to watch about energy before you come on Wednesday. You're going to need that as some background so we can talk a little bit about energy. All right? And that information, the note packet, there is already a note packet 3. I think I have an updated version of that, so I got to update the version, note packet 3, put it up, and you're going to use those videos to sort of fill in a couple of those pages. All right? Cool. Thank you. Awesome. Don't forget, quiz 2 is live until 10 o'clock tonight. And as always, we're recording. Yo, you're good. As always. Hey, was that active or passive? Was that active transport or passive transport? Well, you kind of hit it, but it moved from high to low, so that's passive. Oh, hold on a second. Let me stop the share.
I sent it like last week, actually.
Okay. Let me